For the last 2 years, we and many others have been working on developing PARP inhibitors as a precision medicine drug for patients with advanced prostate cancer. And actually, it has been the first drug class approved for a genomically defined subset of patients with prostate cancers. In the last few years, we have also seen data suggesting that when you combine PARP inhibitors with other drugs, such as hormonal therapies, there may be the possibility of expanding the population that benefits, but there has always been a concern about the lack of mechanistic validation in patient samples for such combination. Thus, we decided to launch ZZ First as a clinical trial that would, on the one side, explore the clinical activity of such combination in an earlier stage of the disease, in line with other trials that were being launched in this population, but also critically as a trial that would interrogate the mechanism behind the potential synergy or additive effect of these two families of drugs, PARP inhibitors, here represented by Talazoparib, and AR inhibition, here represented by Enzalutamide. So ZZ First is an investigator-initiated study that wants to test the combination of Talazoparib and Enzalutamide in men with metastatic hormone-naive prostate cancers, meaning men that have not received any therapy for metastatic disease yet, and that invested severely in acquiring on-treatment samples to investigate the biology interactions between these two drugs. We have now seen 3 different clinical trials combining PARP inhibitors and AR inhibitors in the metastatic castration-resistant disease. So that means in patients that have already progressed to hormonal therapy. However, the hormonal therapies and the androgen receptor inhibitors have now been moved to an earlier stage of the disease, to the metastatic hormone-naive setting, the first line of therapy. Thus, it makes sense to test these combinations earlier, because at present, most of the patients who arrive to the CRPC stage have already progressed to an AR inhibitor. Hence, if there was to be a biological interaction, it would be at the maximum potency at the first exposure to hormonal therapy. In parallel to ZZ First, other larger randomized trials have explored these combinations of drugs in this same setting, but just focusing on the clinical activity. The added value of ZZ First was, on the one side, to expand the population that is undergoing testing, but also acquiring these samples that would enable us to investigate the biological interaction between the drugs. In the ZZ First study, we recruited men with metastatic hormone-naive prostate cancer, so with disease outside of the prostate in other organs who have not yet received any hormonal therapy, and that were considered what we call high-volume disease cases, meaning cases that have a lot of bone metastasis or metastasis in the viscera, liver or lung. That is something quite uncommon in prostate cancer. This is a population that we know that unfortunately has a very poor prognosis with the standard of care options. So we thought that if there was a population who could potentially benefit from a more intense therapy regime, would be those patients with a high-volume disease. Those patients would start treatment with an hormonal combination, namely androgen deprivation therapy and an IR inhibitor, in this case enzalutamide, and then after 2 months of therapy, they would be randomized to the addition of Talazoparib versus continuing just with the 2 hormonal therapies that is the standard of care. Critically, we would implement a biopsy acquisition time point before the randomization, so we would see how the tumor was adapting to the hormonal therapy before adding the Talazoparib. After the randomizations, patients would continue the treatment for as long as they would be benefiting or that they were tolerating the drug. So we would stop treatment if there is progression to the castration-resistant phase or if there is toxicity that prevents the patient from continuing on trial. In this trial, we didn't select patients up front based on any molecular biomarker, but we retrospectively tested the molecular profile of all patients in order to balance the 2 groups for those critical mutations that condition Talazoparib sensitivity, namely mutations in the homologous recombination gene repair and particularly in the BRCA genes. The ZZ First trial recruited between 2020 and 2022. These are patients that normally benefit from this hormonal therapy between 2 to 6 years. There is a lot of heterogeneity, so we've been following up these patients for a number of years now. In 2024, we presented just very briefly results of the primary endpoint analysis, that is how many patients actually are responding very well after 1 year of therapy, and now we are disclosing the whole data set that includes all the long-term follow-up efficacy analysis, so how long did it take for the patients and the diseases to progress again, the toxicity and tolerability data in the long term, and critically also the biological analysis in the samples collected from these patients, both before starting the therapy and the biopsies collected while they were on therapy.